So I'm going to present you a new range of vaccine, synthetic vaccine, organic synthetic vaccine, and we have a first application on HIV.
Organic Vaccines is introducing an innovative, therapeutic, and preventive vaccine against the human immunodeficiency virus, HIV. Organic Vaccines regroup scientists and entrepreneurs who believe the vaccine industry is at a disruptive stage, with vast areas of discovery and opportunities. Among the projects of Organic Vaccines is HIVAX, a vaccine candidate to prevent HIV infection and cure infected patients. The vaccine has already undergone Phase 1 and Phase 2A studies under a French regulatory scheme and is authorized to proceed with further tests. 4 years after initial injection of the vaccine, analysis of volunteers' RNA, DNA, and antibodies may prove some of the patients have undergone some healing process. Our scientific team has gathered some world-renowned specialists in immunology. Organic Vaccines, or a newly created company it may designate, is seeking Series A financing to fund further studies and trials on the vaccine. This new vaccine may lead the way to other synthetic vaccines against influenza, for instance. Organic Vaccines was founded by Patrick Rambaud, former McKinsey consultant and former president of IMS Health France. Erwan Loret is the inventor of the TAT-OI vaccine. He is a researcher with the French National Center for Scientific Research. Professor Dominique Charon is an internationally well-known immunologist, professor of immunology at the Saint-Louis Hospital in Paris, and head of the Jean Dausset Institute. Claude Sarbat is a seasoned pharmacist and former head of several medtech companies in France. Thierry Detloff is a former CFO of several listed companies and non-exec director of several companies. Bruce Butler is co-head of the Scientific Advisory Board of Organic Vaccines, a well-known immunologist and Nobel Prize recipient in 2011. Bruce's work focus is presently on a specific organic adjuvant. For 30 years, no cure has been found against HIV. 37 million patients are infected on all continents, and the infection grows every year by a net figure of 1.1 million patients. Scientists have developed over the years combined antiretroviral treatments, abbreviated as CART, to lower the level of the virus, sometimes at undetectable levels. Those treatments cannot be stopped or the HIV virus will rebound. Antiretroviral treatments have severe secondary effects, and the long-term effects of such treatments are unknown. It is believed life expectancy is materially reduced. Until now, no explanation has been found why antiretroviral treatments do not cure HIV infections. Similarly, there is no explanation why the human immune system does not eliminate the virus. There have been many attempts to develop a vaccine against HIV. Some of them have undergone human trials, but no vaccine attempt has, until now, reached such promising results as the HIVAX TAT-OI vaccine.
And we are planning to do a publication early next year, which I believe will be a breakthrough.
The TAT-OI vaccine is a synthetic vaccine made of amino acids. The discovery of the vaccine came from a study of a population of Gabonese pregnant women who were initially HIV infected and who became seronegative from 1986 to 1988, several months after the infection. Dr. Erwan Loret, the inventor of the vaccine, identified a TAT-OI protein. OI is the name of one of the Gabonese women that could be effective as a vaccine against HIV. The TAT-OI vaccine gives the capacity to have neutralizing antibodies against different TAT variants that drastically enhance the viral transcription. Tests on animals, rabbits, and macaques have been performed with positive results that were publicized. The Berlin patient is the only HIV-infected patient to have ever been cured from AIDS since the virus has been identified. He was cured under very specific circumstances through a stem cell transplant and white blood cell transfusion. His case is scientifically interesting because it shows the healing process from HIV over time, the disappearance of HIV DNA, followed by the disappearance of HIV antibodies. As shown in the timeline, the vaccine process took several years. The Berlin patient is still alive and no longer follows any treatment against HIV. A double-blind randomized phase 1/2A trial has been conducted with the authorization of the French ANSM, French National Security for Drugs, as early as 2013. It covered a cohort of 48 volunteers with different dosages of the HIVAX-TAT-OE vaccine. The trial included CART interruption. 2 years after the injection, 2 patients started to show signs of retro-seroconversion, similar to the Berlin patient, as explained earlier. The study was published in 2016. 4 years later, an undisclosed number of patients among the cohort who have received the adequate vaccine dosage remain at undetectable levels of HIV DNA. It may be an indication they have also started a retro-seroconversion process. This has still to be officially confirmed through an authorized study for which approval has been given. We would like to have an NDA agreement signed to provide further information due to the sensitivity of this information. Undetectable HIV RNA and DNA do not represent a total confirmation that the patient has been cured from HIV and that the specific HIV reservoirs are no longer infected. As demonstrated by the Berlin patient, specific HIV antibodies should also decline and disappear. The above tables show the evolution of the most specific HIV antibodies over time for the 2 patients mentioned in the 2016 publication in Retrovirology.
So that's the first time we are able to make the HIV DNA undetectable. So that's the first time, and we are even looking for a different type of analysis in order to really go beyond 2 or 3, below 2 or 3 copies of HIV DNA. Today, we measure the HIV DNA with tests which are measuring only 20 copies. So it's really a world premiere.
No other HIV vaccine attempt has reached such positive signs as the one shown by the phase 1/2A study for the TAT-OE vaccine. The roadmap to finalize the HIVAX-TAT-OE vaccine includes an official follow-up of the phase 1/2A volunteers to confirm their level of HIV DNA and HIV-specific antibodies. If signs of retro-seroconversion are confirmed by this follow-up study, a double-blind phase 2B trial on 150 patients will be conducted in the course of 2019. The timetable for such studies should be relatively short, with a first milestone once the follow-up study has been conducted. Organic Vaccines plans to enter into alliances with larger pharmaceutical groups once phase 2B preliminary results will be available. Additional intellectual developments for the HIVAX vaccine include adjuvanting the vaccine, developing and patenting a closed manufacturing system, developing a nanosynthetic vaccine for nasal spray injection. In the future, the technology of the HIVAX-TAT-OE vaccine could be applied to a synthetic vaccine against influenza. Intellectual property of the HIVAX-TAT-OE vaccine is as follows. 2 initial patents have been registered by the French National Center for Scientific Research and licensed to a company that is presently no longer active in the development of a vaccine for financial reasons. Organic Vaccines is the beneficiary of an exclusive mandate from the licensee to develop and organize distribution of the vaccine. Organic Vaccines will also develop a nano vaccine allowing nasal injection. Organic Vaccine will also test and implement the vaccine adjuvant, possibly using the organic adjuvant developed by Professor Bruce Butler. Target markets for an HIV vaccine are huge, as combined antiretroviral treatments, CART, are presently very expensive. The HIVAX TAT-OE vaccine would probably not replace all CART, nor reach the same level of sales of today's market, in the range of $20 billion US. It would replace some of that market and also cover all non-infected population. A low-cost vaccine, probably in the form of a nasal spray, would also be needed for underdeveloped countries. If successful, the vaccine would be urgently granted a temporary use authorization that would generate sale immediately at the end of Phase 2B trials. The company does not intend to release any sales forecast. Total expected cost of the project until regulatory market authorizations and building of a production facility amount to 27.5 million euros. Organic Vaccines is raising Series A financing for an amount of 5.5 million euros. Such financing should bring development of the vaccine until end of Phase 2, thus enabling agreements with pharmaceutical companies. At this stage, an estimate of 10 million euros have already been spent in R&D, trials, and equipment.
Mostly with grants from Madame Betancourt, from US donors.
This table illustrates how Series A financing will be used in the development of the vaccine, for a total of 5.5 million euros.
So if you have any question, I will be happy to answer to anyone.